crutches like that without them falling over is a real skill. Takes years. Thank you very much, Chris. Thank you. And hello to everyone here today. First of all, I would just like to thank my predecessor, Cheryl Scudamore, for everything she's done for the fellowship over the past few years. And secondly, I'd like to sincerely apologise to all the RCVS staff who thought they had got rid of me. And here I am, back again. In all seriousness, I went for this role because I can see the fellowship is already making a very valuable contribution to our veterinary and indeed veterinary nursing professions. I also believe there's a great potential to do much more in terms of increasing public understanding around the art and science of veterinary medicine and providing inspiration to mid-career veterinary surgeons who are maybe looking for fresh challenges in becoming part of this learned society. As former chair of the RCVS Diversity and Inclusion Working Group, I'll also be focused on widening participation within the fellowship, not only looking at how we can get a more representative and balanced membership, but also how the fellowship can, through its work, reach out to a greater variety of people. On the subject of outreach, our Fellows of the Futures competition is a great example of how the fellowship is having an impact, bringing together different generations of veterinary surgeons to talk about new and exciting research. And although we will just be having 3 presentations here today, as Chris said, the number of entrants we've had overall was actually 38, which just shows what a crucial platform Fellowship Day and this competition is. Regarding the competition itself, one of the fellowship's remits is to use its accumulated knowledge and reach to inspire all parts of the profession, including those currently studying veterinary medicine. To meet this aim, the Fellows of the Futures competition was established to encourage students and graduates currently involved in research projects to share their work. Undergraduate and postgraduate students and recent veterinary graduates on the RCVS register are all invited to enter. And as I said, this year saw 38 applications submitted, and the judges have shortlisted the top 3 to speak here today. So each student has 5 minutes to present their research and a further 2 minutes to answer questions from our panel of judges. Entries will be marked against a number of criteria, including originality, contribution to scientific knowledge and understanding, knowledge of the subject material and quality of the presentation and delivery. A £200 National Book Tokens gift card will be awarded to the student who is judged to give the best presentation. The presenters of the 2 highly commended presentations will each receive £100 voucher, and obviously all 3 of you are winners straight away to get here. So, and I would like now to invite on stage the 3 judges for the competition, Rachel Jean, Elvin Kilendra and Pam Mosdale, just to say hello. Or you can even just stand up. Yeah, that's fine. And I would just like to say that Jade Stat, who's done terrific work assessing the actual entries that are coming in, has done terrific work, but she sends her apologies, and I hope she feels better very soon. So we're going to actually just kick off straight away. And are going to introduce our first finalist, Albert Cliff from the University of Cambridge, who will be presenting his work on detection of avian influenza virus in wild birds to determine prevalence, subtype, and fitness cost of infection. Okay, Albert, fire away. I'll get you away.
Hello, my name is Bertie. I'm a 4th year vet student at Cambridge, and I just wanted to give you a brief overview of the research I carried out last year. Avian influenza virus is a small RNA virus which has the capability to cause highly contagious respiratory and gastrointestinal disease in birds, along with the zoonotic potential to spread to us. Waterfowl species, a type of wild bird, act as reservoirs for the disease, and it's their incursion into domestic poultry flocks which creates a significant risk to bird health and welfare, but also economic repercussions that have knock-on effects to society. So the aim of my project was to fine-tune a nucleic acid extraction method from avian fecal samples acquired from multiple locations across the UK. This genomic material was then used in detection and subtyping PCR assays, along with determining other factors such as co-infections and any fitness cost to infection. Along with obtaining this information, it was hoped that the project could demonstrate a proof of principle for a wider scaled method of testing in wild birds, which would help which help positively promote disease surveillance and prevent incursion of the virus into domestic poultry flocks. So on to some of the preliminary results. Using primers for a conserved part of the matrix genome segment of the virus, avian influenza was detected in endpoint PCR assays and qPCR assays, which the qPCR are unfortunately not shown here. This was in a multitude of samples, including barnacle geese from the Solway, Scotland, between March and April 2022, and also redshank and oystercatcher samples from the Wash between August and September 2022. Further to this, subtyping of neuraminidase and hemagglutinin segments, back there still, revealed a variety of subtypes, including high pathogenicity forms, H5 and H7. However, subtyping was not as effective as I would have liked, and that stems back to the complexity of the samples obtained. Now, a significant finding was that a decrease in weight when standardized to head-bill length ratio was found in a particular population of redshank that had tested positive for the virus, compared to redshank in that same population that had tested negative. Now, we were able to conclude this as the redshank and oystercatcher samples from the Wash were associated with specific birds. When these birds were caught during routine ringing practices that take place across the UK, their weights and their sizes were measured, and if that bird defecated, that sample could be collected and assigned to that specific bird. The fecal samples used in this research were non-invasive, giving them particular advantages over other modes of collection, such as cloacal swabs or oropharyngeal swabs. The same protocol developed in the research was also used to identify a variety of other organisms that may be present within the bird, either as commensals or as pathogens. Eimeria species themselves were identified in numerous AIV-positive and AIV-negative birds. Eimeria itself is a significant risk to the domestic poultry setting, so it's the ability to simultaneously test for other pathogens in these samples, which can positively promote disease surveillance, identify any local reservoirs, and help prevent incursion into domestic flocks. It's important to note that the fecal samples used in this project had issues. They were poo at the end of the day, so contained contaminants from the bird's own commensal flora, the food that it's eaten. However, they are non-invasive to the bird, potentially allowing for a much larger scale and capacity of testing, which can be done on wild birds. They could just be picked up from the floor or taken during routine practices, such that of bird ringing. So the hopes of my research were to demonstrate a reliable nucleic acid extraction method from avian fecal samples to be used in detection, subtyping, and potentially even sequencing assays. The more birds across different populations that can be tested for the virus, the more effective disease surveillance could become. Thank you very much, and I'd like to quickly thank Dr. Anna Patasio and everyone at the lab in the Department of Pathology for your amazing support during my project. Any questions?
Thanks so much, Albert. That was terrific.
Thank you.
Have we got any questions from the panel for Albert?
Albert, I had some pet chickens until very recently when unfortunately the fox got them. But how would you envisage this being actually used for poultry owners?
So obviously it would maybe have a more positive effect on free-range flocks or backyard chickens, where if you are testing on a scale almost similar to that of COVID testing that we had, we can identify local disease reservoirs within a day or two. These samples can be shipped off in the samples, tested and subtyped in the same day. And if there seems to be a higher prevalence or subtypes of note in that local area, then methods of biosecurity then could potentially be put in place because of that.
So it could just be local lockdowns rather than...
Yeah, exactly. So instead of kind of widespread concern, we can limit lockdowns and biosecurity measures to local disease reservoirs. Yeah.
Would there be any way of using these samples for genotyping the birds from which they came? Are there enough epithelial and other tissues to do that?
Potentially. So that was one of the issues that I faced. I did attempt to to sequence the virus within. However, obviously a lot of these samples had degenerated. That's the nature of the fecal sample. So obviously with other modes of collection such as cloacal or oropharyngeal swabs, then it might be easier to subtype because there's probably less contamination.
What about the genotype of the bird?
So the genotype of the bird, as I would say, so probably with that, the contamination, the number of epithelial cells or the bird's own DNA probably would be harder to get from those fecal samples. So that's when probably those other forms of collection would be more beneficial for that.
Fantastic presentation, Albert. Thank you so much. I was just wondering whether how your novel testing method would compare to more traditional methods in terms of the speed of results and also the cost as well.
So quite simple sort of method. The samples were collected in RNA-DNA shield, which inactivates the virus but keeps the viral, the DNA and the RNA within that sample. It conserves it. So these samples were posted. They can be posted at room temperature, can be posted first class perhaps, received to the lab the next day, and PCR assays don't take too long. And now with the qPCR methods, which weren't shown here but could be used, it takes only a few hours really. So there's a potential for quite fast testing.
And is it commercially available yet?
So the DNA kits are available. There's obviously extraction methods for nucleic acid in cells, and samples have been around for some time. So it's developing method. We had to alter ratios of product to make sure we're not degrading too much RNA, but trying to get rid of all those contaminants there's going to be thousands of bacteria within those birds that have also got much more genomic material than these small RNA viruses. So it was all about whether we were actually reduced our rate of homogenization, for example, so we weren't actually breaking down big bacterial cells and epithelial cells with their big cell membranes and cell walls, whereas we were just breaking down the smaller particles such as viruses.
Thank you very much. That was terrific. And, honestly, you answered those questions really well, and that was a great presentation. Thanks a lot, Albert.
Thank you very much.
Superb. Okay, so our next presentation is, our next finalist is Daisy Cookson from the University of Liverpool, and she'll be presenting her work on the biological response of canine cruciate ligamentocytes following treatment with synovial fluid extracellular vesicles. Thank you very much.
Hi, so I'm Daisy. I'm a final year student at Liverpool, and I completed an intercalated Masters of Research, where I looked at cruciate ligament disease and extracellular vesicles. So I thought I'd just start off with a little bit of a background. So as we know, the cruciate ligament is a really important structure in the canine knee joint, and we know that cruciate ligament disease is one of the most common differential diagnoses for dogs with pelvic limb lameness. So it's extremely prevalent and extremely debilitating as it expedites osteoarthritis progression. However, despite the prevalence, the exact etiopathogenesis of cruciate ligament disease remains undefined, along with the crosstalk that occurs between joint tissues. Furthermore, although we know that certain breeds are more predisposed, such as Labradors, there aren't any diagnostic tests capable of identifying at-risk individuals, and there aren't any treatment options that target prevention, with treatment often being limited to surgery. Within my project, I looked at extracellular vesicles, and these are double-layer lipid-bound vesicles released by all cells into the extracellular space. And within EVs is their cargo, and this includes small non-coding RNAs, such as microRNAs. And we know that both EVs and their cargo are a key component of cellular communication. Within the joint, EVs and their cargo have been shown to play a role in inflammation by inducing the release of inflammatory mediators, inducing catabolic changes, and contributing to degradation of the extracellular matrix. Furthermore, imbalances in EVs and their cargo have been shown to contribute to degradation and joint destruction, but their role in ligament disease has not been fully explored. So within my project, I had two main aims, the first of which being to characterize the synovial fluid extracellular vesicle population, which we isolated from canine stifle joints with and without cruciate ligament disease, so healthy and diseased synovial fluid. And then my second aim was to identify the response of ruptured cranial-cruciate ligament cells, which were collected from dogs undergoing TPLO surgery, to incubation with the EVs that I isolated in my first aim, to identify whether there's any difference between treatment with the diseased EVs versus treatment with the normal EVs. With our hypothesis being that as the cruciate ligament degenerates, this then leads to release of extracellular vesicles into the synovial fluid, which contain altered cargo, further dysregulating the knee joint and therefore leading to and contributing to ligament rupture and acceleration of osteoarthritis. So in terms of my results, in the first aim, I first of all found that the EVs isolated from joints with cruciate ligament disease were both larger and more abundant. Unfortunately, we were very limited with a sample size, and so I couldn't perform any stats on this, and so we'd obviously need further studies to validate these findings. I also found that the microRNAs from healthy and diseased synovial fluid were distinctly grouped, as can be seen in the PCA plot, and we identified 57 differentially expressed microRNAs. So obviously this is very preliminary, but in the future this poses a question as whether we could use EVs and other molecules within the synovial fluid, such as microRNAs, to identify at-risk individuals, to allow earlier intervention, or as biomarkers to indicate disease progression. In terms of my second aim, of most significance, I found that the diseased EVs caused a significantly higher relative expression of SOX9 in the ruptured ligamentocytes, but compared to both the control and the treatment with normal EVs. SOX9 is a marker of chondrocyte differentiation, and it's a key chondrogenic transcription factor. And across the literature, it's been shown that as ligaments age and as they degenerate, levels of SOX9 actually increase, and this reflects the change in ligament cell phenotype to a more chondrocytic-like cell, which can be associated with a change in ligament biomechanics. So this increase in SOX9 is a recognized pathological change in ligaments. And so again, this is all preliminary, but in the future, this again poses a question of whether we could use EVs to further our understanding of the etiopathogenesis of cruciate ligament disease and help us to understand the crosstalk that occurs between joint tissues and therefore potentially provide us with alternative therapeutic strategies. So I'd just like to thank my wonderful supervisors, Professor Ethna Comerford, Professor Mandy Peffers, and Dr. Yauda Ashraf-Karaz, and thank you for the opportunity to present my research, and I'd be happy to take any questions.
Thank you very much, Daisy. I think there are probably a number of orthopods in the audience similar to me thinking, please don't take away our TPLOs. Or please do. I guess my first question to you would be, did you look at any breed variations in the vesicle population?
Yeah, so it is something we did collect. So we obviously did collect the breeds. Unfortunately, we were a little bit limited with sample size, and so we weren't able to actually compare the breeds, but I think that's something that we definitely would like to look at in the future. But we did just have like a pooled sample of the synovial fluid because we were quite limited.
Fantastic. And then I guess following on to that question, I guess your hypothesis was whether there was degeneration in the ligament which was causing the vesicles in the synovial fluid. Could you see that there would be another reason why there would be an increase in the vesicles in the synovial fluid, which is then leading to a degeneration in the ligament?
That's a really good question, and something that I don't think I considered, but I would have a look into, yeah.
Fantastic. Just pass it on to my colleagues.
There's a question there.
Yes, I wondered if you've had a chance to look at inflammatory cytokines, which in human beings are very significant factors in degeneration of various joints.
Yeah, so it's not something that I looked at in my project, because I focused on markers of, like more sort of ligament markers, but again, something that we could look at in the future.
Rachel?
Great presentation, well done. As a feline medic, really by trade, nothing to do with TPLOs and all those other horrible abbreviations, I think it would be marvellous if we have less invasive treatments. You talked about if we can understand some of this science more, it might lead to alternative therapies. Had you had any discussion or thought about what we could do to get in the way of some of this?
Yeah, I mean, I guess that's a research project in itself, is looking into whether, you can have an exercise intervention, or whether, later down the line, our EVs and our microRNAs sort of modulating that could be a potential, but yeah, something to look into later down the line.
Any other questions?
Yeah, and similar to Rachel's question, you said about identifying risk. As the owner of a Labrador, what would you do? Advise the owners who do have a risk factor for this?
Yeah, I mean, again, this is a really good question. I guess just being aware of what to look out for, and yeah, I'm not too.
Maybe keep the weight down.
Keep the weight down. Yes, keep the weight down. But yeah.
Thank you.
Thank you.
It was brilliant. Thank you.
Thank you.
I was just going to ask you, just out of interest. I mean, obviously, most cruciate ruptures occur as a result of degenerative changes. Do you know if there was any traumatic injury in part of your sample?
Not that I'm aware of,
okay.
But yeah.
Okay, right. That was terrific.
Thank you
very much. Thank you. Right, that was tremendous, Daisy. Thanks very much. So now we're going to invite up Ellie, and Ellie's from the University of Nottingham, and she's going to present her research on the recognition of pain in calves and decisions around treatment among farmers in the UK. Thank you very much.
Hi there, nice to speak to you all today, and I want to say well done to my two previous students. You've given me quite a hard one to follow here. So my name's Ellie, and I'm a Nottingham 5th year as of today, and I'm here to present to you my project funded by the Animal Welfare Foundation. So we know calf disease is a big problem, with mortality rates in the first 3 months of life being 5%. Data from 2021, as shown on the graph here, shows that the issues faced by British farmers when it comes to respiratory disease and diarrhoea. It's recognised that non-steroidal anti-inflammatories improve our outcomes by decreasing pyrexia and increasing intakes, by reducing pain, and ultimately we end up with happier animals which get better sooner. So let's get into my research. We needed to determine what is a sick calf on farm and how farmers respond to this, and when sick calves are perceived as in pain. So our aim was to determine the impacts, the decisions of treatment of sick calves on farm. To achieve this aim, we used a mixed methods approach. Firstly, we used an online survey which asked the respondents how they routinely used NSAIDs in calf disease, and the results of this are depicted on the graph on screen, with the UK highlighted in bold. And unfortunately, we had a disappointing figure that less than 50% would use NSAIDs in the response to treatment of diarrhoea, although when it came to respiratory disease, it was slightly better. In addition to the data shown on screen, our survey also included 8 separate calf scenarios, which are shown through a video. We then asked our respondents if they'd use NSAIDs, electrolytes or antibiotics, and to rank this on a like-it scale. We were astounded by the results we received. We received 896 viable responses from vets, farmers and consultants globally. And it was really interesting to see that our farmers had a wide range of treatment decisions when it came to each of these 8 scenarios. Interestingly, our vets that answered were very static, although the scenarios differed. And it was this variation we discussed further in our focus groups, which were carried on Microsoft Teams. And this is where I really took the lead, by taking the lead on the focus groups and additionally analysing the data through thematic analysis. So, what did we find in our focus groups? Our focus groups revealed 2 key themes: the motivators and the barriers, which ultimately impacted the clinical signs seen and therefore the decision to use NSAIDs in a sick calf or not. And our key barrier we found was education. And I think this is an area we can make a massive difference, because ultimately, as vets, one of our roles is to portray the new and upcoming research to our clients. So, we can break this barrier of education down into 4 key areas. Improving knowledge. It was felt that there was a lack of industry drive and support and a lack of drive to improve farmers' knowledge, as demonstrated by the quotes on screen. I do hope you can read those at the back. And stewardship. Through this research, it was identified that there was an area of confusion between how we use an antibiotic and a non-steroidal anti-inflammatory drug and how they were different. And this level of ambiguity I think we need to solve. Decision making. It was all about empowering the farm staff on the ground with the right knowledge, the right tools to make the right decisions. And finally, awareness of effect. This is the concept surrounding when to implement NSAIDs on farm in our treatment protocols, how they work, and crucially, why we should be using them. So in conclusion, our farmers are very, very passionate about their calves and really want to prioritise them. However, there is the boundary of education which stands in their way of making informed decisions. I'm personally passionate about education, and I put to you as an industry, we need to find more accessible ways to educate and engage our farmers to help promote animal welfare and ensuring productivity and efficiency on our farms. So thank you for listening to me today. If you'd like to come and chat with me at lunch, I'm more than happy to. I'm also happy to take questions from the floor now.
That was epic, well done.
Thank you.
It's great to see someone else caring about what our farmers think and caring about calves. You mentioned there as one of your quotes some confusion between antibiotics and NSAIDs. Can you elaborate on that?
Yes, so this was all gathered through our focus groups. And I think, as being a dyslexic individual myself, it might be just, they both have anti in them. And it appears that there is a massive drive from us lot that we want to reduce our antibiotics, which I completely get. However, some farmers, I think, have taken this of reduce buying stuff from the vets, which includes anti-inflammatories. So instead of just decreasing their antibiotic use, they're decreasing their drug use, which is good, but at the end of the day, I always see this as, if I've got a headache, I take a paracetamol. So why on earth are you leaving a calf with scour or with respiratory disease without what effect? It's, you know, it's paracetamol for calves. It's not, but it's a really good way of putting that point across.
It's really cool. Language is so important around these things. So do you think we have a good evidence base at the moment that these drugs will improve outcomes for calves?
I think it's coming. I think when we spoke to our farmers, there are some quotes up there, and they can tell you can see the difference. And it was all about, and we believe, get in there early. As soon as it becomes ill, if we give it some pain relief, it probably doesn't become as ill. And I'm not too sure, definitely when it comes to our routine management, we see a lot more data behind this. This is why we looked at sick calves, but you can definitely see it on the ground, and the anecdotal evidence is definitely there, and farmers were portraying this when we were asking them in these focus groups.
Yeah, that was brilliant. It's a long time since I've been in mixed practice, and then probably we didn't use pain relief anything like enough. But I was really fascinated by all the videos, of all the idea of the videos of the different calves. I don't know, maybe there is, but I know there's validated pain scoring in small animals and horses. Is there anything like that for calves?
Not too sure, but we do have the grimace scale, which we have in various animals, and it doesn't take a leap of imagination to jump between species. And it was really interesting. So our 8 videos were just videos. There wasn't history behind them, which we did give in the focus group, but we didn't give for this online survey. And these videos had respiratory, so like watery eyes, obvious scour or just a little bit. We also had one what we considered to be normal. The respondents did not take that calf as normal, and that really shocked us. And it was really interesting. Unfortunately, I don't have the graph with me today, but our farmers, like what they'd use would go up and down depending on the 8 different scenarios. Our vets, I could have taken the one bar and copied and pasted it over.
Really interesting.
We are still looking at that. That data analysis is still ongoing, but from that initial graph, it's really interesting to see how potentially what we look at for a sick calf on farm and the clinical signs we notice are very different to that of our farmers, which again came out through our focus groups when I asked them, what can you tell me about this calf?
So that really gives you some potential for maybe guidelines around, for the farmers around
Yeah.
what to look for.
Sorry, just one more question, Ellie. And just to say, it's very humbling to see what amazing work all of the Fellows of the Future are doing at such a young and early age in their career compared to, I think, what the majority of the audience were doing at your stage. So huge congratulations to everything that you've done.
Thank you.
And also, I think it's also fantastic how you've identified so early on that education is key to the population. So going on from that, did you notice any differences in cultural views based on the geography of where you obtained your data, and how you would potentially tailor-made your education depending on who you were looking at and where they were coming from?
Kind of, yes. It's definitely still part of the analysis which is ongoing. As in our survey, we collected demographic data, which we grouped into like UK-based, Europe or America, although in our focus group we only asked British farmers. And I think truthfully, ultimately, a lot of the farmers did not want this top-down, they wanted this bottom-up. And I think all of you can tell me today, you all know your clients and your environment so much better than the board of people sat in an office. I think we really need to empower our vets on the ground, but also the person at the tractor merchant who sells them like hay, straw, and all these like SOPs and other people in the industry that the farmers listen to, because I'm sure we can shout down their ear for 10 years to do something, but as soon as Joe down the road does it, it gets done. So we need to work as an industry together, and it cannot come just from us. We need to talk to everybody involved and get us all singing from the same hymn sheet here.
I think we've got time for Frida. Were you going to ask a question? I think we'll make this the last one.
I'd love to ask a question, if you don't mind. I sit here as a vet and a farmer, an organic farmer, and for years we've been using non-steroidal antibiotics. Because, thank you very much, that will save my voice. My name's Frida Scott Park, and I'd love to talk to you further. You've made a comment there that I can just read. The calf's health and comfort should come first, and the actual cost of not the actual cost of the treatment. But I have to say that the cost of the treatment can be substantial, and I wonder if any future studies, or whether you looked at the economic status of each farm and their use of non-steroidals on it.
We didn't look at it directly, although in our focus group, because when I came into this study, it was an area I thought was going to come up. Not one person mentioned it when I asked them directly, what are your barriers to using NSAIDs? Not one person mentioned the cost of the drug. The only place the cost of the drug came out was actually the motivators. But what did come out of the study, and I think there's a quote on screen there, I'm sorry for the guys at the back, was actually, it's not the cost of the drug directly, It's these calves aren't your milking herd, so they're not making you money now, although they are going to make you money in the future. Or going up the ladder, if you're selling to beef at an auction, they don't mention that you've put all this time and effort in and it's had its non-steroidal nice and early. We don't mention that. There's no badge to say, look how good I care for my calves, which would then increase the value. So it's not the cost of the drug that's impacting people, it's the cost down the lines and all the other factors of the cost rather than handing over the money for the bottle, if that makes sense.
Thank you very much. That
Thank
was
you.
fantastic. Thank you. Well, how good was that? 3 outstanding presentations. And really informed, very pertinent, right? A fantastic species spread. And I think, if we take these 3 young people as examples, I think we can safely say our professions are in good hands. Thank you very much indeed.